Bacteria are evolving to escape antibiotic treatments, and at some point we will not have any antibiotics available that can treat those infections. One example of how bacteria can inactivate antibiotics is the expression of certain proteins, and these proteins can break down the antibiotics. We have developed a new approach which is based on a metal complex. With light irradiation, our metal complex will specifically damage proteins which are responsible for drug resistance.
My name's Lars and I'm a research scientist. I'm going to show you how our metal complex that we've made works. So just pretend that this table here is the inside of our E. coli bacteria. Now you can see this is the protein that's evolved to break down antibiotics. The protein cleaves the antibiotic so that it's unable to kill the bacteria. This is the molecule that we've designed. It consists of two main parts. This part is the ligand. So this binds to a specific part of our protein. This part here is the metal complex. Once the ligand binds, it brings the metal complex close to the protein so that it's in the perfect position of what's going to happen next. Once activated by blue light, this metal complex produces highly reactive oxygen molecules called ROS, which cause damage in the immediate area. Once the blue light has been turned off, our metal complex stops producing ROS and is now released from the protein. Now our molecule is free to move on to the next target protein. With more of these proteins gone, this makes our antibiotic more effective. Now I'm going to show you how we check that it works. So the metal complex looks like this. It's an orange powder. We dissolve this complex to make this solution here, and then what we're going to do is basically dilute this into this plate here so that we have a high concentration of metal complex along one edge of the plate, and then this is diluted down this edge of the plate. And then in this plate we have the antibiotic. What happens is we then combine these into a third plate. These concentrations will then allow us to create a checkerboard-like assay where we have a different combination of metal complex and antibiotic in each of the wells. So using different concentrations allows us to test whether a higher concentration of our metal complex has a greater impact on the effect of the antibiotic. Because our complexes are activated by blue light, and this light has some blue light in it, we don't want the complex to be activated when it meets the bacteria until later. It means we have to do this experiment under red light. We're ready to mix our compound and antibiotic with our bacteria. We now let this plate incubate to give some time for the metal complex to enter the bacteria. So this light is activating our metal complex to degrade this protein that is the cause of the resistance in the bacteria. After the bacteria has been growing for about 24 hours, we need to measure this. So this machine here measures bacterial growth, and so you can see here that at higher concentrations we see no bacterial growth, especially when irradiated with light, compared to when we do this in the dark. It's rescuing the activity of this antibiotic.
Our approach is based on a metal complex in combination with light treatment. And as we need light, we need to be mindful of what bacterial infections we can actually treat. One example could be if you have a surface or wound scratch, you could take an antibiotic and our metal complex could be spread on that wound, and with light irradiation you could then treat that drug-resistant infection. This is just a first and initial approach, and when we have shown that this actually works. In the next steps are to also look at the chemical structures and maybe also use different wavelengths of light, that we would have deeper tissue penetration so we could affect different infections in the body.